Welcome to this question and answer session about unbiased stereology. My name's Dan Peruzzi. I'm a research liaison and head of technical support here at MBF Bioscience. MBF Bioscience is sponsoring this webinar. Stereology is the study of solids. When we as scientists want to look at tissue at a microscopic level and study the solids that make up the tissue, we want to do it without making mistakes. So that's why the name of today's webinar is Unbiased Stereology. We have over 100 people out there in the audience. Thanks a lot. It's really great to be together with you today. Thanks also for the questions that you sent in. I took the first 30 questions that were sent in and organized them by category. These are the categories we're going to look at: unbiased stereology overview, and then sampling and imaging, and then estimating number. So please send questions that pertain to the current category as I'm talking about one of these three categories. This is how you submit questions. You can click the orange arrow to expand the window, and then just find the question tool panel. That can be undocked if you want it to be bigger, and type your questions in. Okay, let's get started with the first category. We're going to do a succinct overview of what can you do with unbiased stereology. And as I said, we have questions sent in from all over the world. Matthew from Canada writes, "I am an incoming master's student looking to learn what I can about stereology. I appreciate the enthusiasm, Matthew, with the exclamation point. Shruti from India is expecting an overview. Emily from the United States is attending for a refresher. Sylvia from Italy and Chad from the United States also want a refresher or an overview, and both of them mention a specific region of interest, the hippocampus." So if I can impress one thing on you today, that is that we need to use systematic random sampling when we are sampling for our unbiased stereology. If possible, we want to sample through the entire region. We want to avoid doing things like using the middle section of the region and assuming that that represents the whole region. Instead, we want to go through the entire region, and we want to use systematic random sampling. On the left, it shows an illustration of systematic random sampling on the section level. And on the right, it shows that when we get the section up on the microscope or however we're imaging it, we probably are not going to look at the whole area. We're going to use a subfraction with the area. So we're going to do systematic random sampling. That's the illustration on the right, is showing systematic random sampling on the section level. Those are the sites where you're going to be restricted to sampling, and there'll be what's called a counting frame or a dissector at those sites. So use systematic random sampling, and we're going to, when we get to the sites that we're sampling, we are going to use a probe to find out about the tissue. The probe is the geometrical shapes that our program called Stereo Investigator is going to generate. It's going to superimpose it on the tissue, and our job is to count the number of probe feature interactions. For instance, if you're estimating number of cells, you may be putting an X on the top of the cell. Or if you're estimating the length of blood vessels, you would be marking where the blood vessel biological string intersects a virtual plane. We want to stick to fractionator type probes if possible. These require that the percent sampled is known and estimates are extrapolated. So this shows what, after we do the systematic random sampling, what can we estimate while we're doing the systematic random sampling. At the bottom, we have pictures from everyday life to kind of let you know that what we can sample with unbiased stereology is volume, surface area, length, and number. So in the cartoon on the top, tissue is shown in green, and the virtual shape that Stereo Investigator will generate, the probe, is shown in red. So moving from left to right, we can use points to probe for volume. We can use lines to probe for surface. We can use planes to probe for biological strings like blood vessels or axons. And on the right, it shows that we need a three-dimensional situation. That's why there's a volume in red shown. That's the dissector. And to estimate the number of cells without bias, we look for the leading edge or the top of the cell and see if it's in the dissector or not. So use systematic random sampling, and we can estimate volume, surface, length, and number at the microscopic level when we're looking at our tissue that we want to find out about. So that was a couple PowerPoint slides. Now I'd like to go to a website called stereology.info. A lot of people have written articles on this website. I have been largely curating this website and writing articles for it. Let's just bring the web browser over to where you can see it. So this is stereology.info, and it's sponsored by MBF Bioscience. This is a great way to find out about unbiased stereology. I want to show you one way to search. Here's the probe index. It's broken down to regions and particles. For instance, if you know the name of the probe, if you know that you want to use the optical fractionator, you can click here. It will bring you to an image of the optical fractionator. It will show that the optical fractionator is to estimate number of cells and that we need thick sections. If I click here, it brings me to guidelines about estimating number, and it tells the formula for the optical fractionator and gives the references. Okay, let's go back to the home page of stereology.info and go back to the probe index. We can also look at probes that estimate surface or volume about particles, and there's one probe called the nucleator, which is a great probe to use with the optical fractionator to estimate the volumes of cells. A lot of times you don't know the name of the probe, but what you do know is what type of data that you want to estimate. So you can click what are you estimating, and this breaks down the probes into first order, which have to do with number, length, surface, and volume, or second order probes, which have to do with the relationship among cells. For instance, are they clumped or are they evenly spread out? First order is broken down into region or particles. So if you know you want to estimate the number in a region, you click here. It tells you all about estimating by number. It shows that there's a probe called the optical fractionator that can be used in thick sections. But if you have thin sections, you can use the physical fractionator. You need pairs of thin sections. Or if you're in a two-dimensional situation like a blood smear or a petri dish, you can use the fractionator probe. What I'm going to do right now is click on the probe index so that we can get an overview of really the most useful probes to use. On the left, again, the tissue is in red. On the left is shown volume. This is a membrane, a biological membrane. It might be like the respiratory membrane in the lung that you're trying to estimate the surface of. These strings in red could be blood vessels or axons, and over here these red circles are cells. So to estimate volume, and let's show the names of these probes, you can use a probe called capillary point counting, and these are the thin sections that you can use. The X's are the points. To estimate surface area or length, we want to use thick sections. So this black rectangle is trying to show that this is a thick section, maybe 30 or 35 microns thick after histological shrinkage. To estimate surface or length, use a thick section. Then you can use an isotropic probe. For surface, we use the isotropic Fokker, which is a triplet of lines that are mutually orthogonal to each other. To estimate length, we can use a sphere. The probe is called Spaceballs. To estimate number, we want to be in a three-dimensional situation, and it's easier to do if you have a thick section and use the optical fractionator. If you don't have thick sections for surface, length, and volume, there are thin section probes that you can use. There's an extra step for these probes, though, in that you need to manipulate your tissue block. For instance, this shows an isotropic section. That means you randomly rotate the tissue block in 3 dimensions. Of course, anatomists don't like that because they can't read the tissue anymore. That's why we want to stick to thick sections for number, optical fractionator, length, Spaceballs, surface, isotropic Fokker. For volume, we can use thin physical or optical sections. So that was stereology.info, and I highly recommend you go there. You can get a lot of questions answered there. Let's go back to these questions. Matthew, Shruti, Emily, Sylvia, and Chad all wanted an overview. In summary, use systematic random sampling. You can estimate number, length, surface, or volume. Sylvia and Chad mentioned a specific region of interest. Really, any region of interest, any type of tissue is fair game for these probes. But if you're working in the hippocampus, you're kind of ahead of the game, because during the history of unbiased stereology, some of the great names that have developed unbiased stereology have been working in the hippocampus. So I would take a look at these references. We see names like Gunderson, West, Slomianka, Schmidt, Peterson here. So take a look at stereology.info and take a look at these references if you're working in the hippocampus. Okay, so that's our first category. And I'd like to pause now. If anybody out there has questions, now is the time to type them in. If you have questions about systematic random sampling or about what can we do with unbiased stereology. Okay, I'm going to move on to the next category. Oh, I am getting some questions here. I have a question from Dermas, and they're asking, I'm interested in length. How can I estimate length of intestinal villi? And you mentioned the isotropic Fracnair probe, but really that probe is to estimate surface area. So to estimate length, you would want to use the probe called Spaceballs. If you contact us, we can help you using that probe. But to estimate length, use the Spaceballs probe. The sphere is a collection of infinite planes arranged isotropically, and what you do is you count the intersections of the villi with that sphere. If you used isotropic Fracnair, then you could get the surface area of the villi. Okay, so Shyam's asking me how to use unbiased stereology for microglia. The same, for that, we want to use the probe called Optical Fractionator. And for our third category today, I'm actually going to show an example of using the Optical Fractionator. And if you're estimating the number of astrocytes, they're like any other particle that you're estimating. You need to find the leading edge, probably of the nucleus you would use for that. Let's look through some of these other questions. I'm getting a lot of questions. Thank you. Okay, this asks, what is the best way to establish the parameters for systematic random sampling? I'm going to go over how to do that later on in this talk. Someone's asking me about second-order probes. I will not be speaking more about those, but we have plenty of questions, and we want to do another question and answer session, so we could get to it then. Okay, let's see if there's questions that are relevant to systematic random sampling or an overview. Okay, here's a question. Do we use live images, or can we acquire images and use them? That's our next category, and the simple answer to that is you can use live images or captured images. There's a question about estimating volume. We will get to that in a future webinar. Okay, someone, Brady has asked, I see people estimating number of cells in the dentate gyrus, but by only assessing the first 5 slices of the hippocampus, is that not a biased approach? That is a biased approach. That will only tell you about the first 5 slices of your region. Instead, we want to use systematic random sampling, where we have an interval. Let's say you have 100 sections through the whole region. Then you might set your interval to 10. You'll do a random starting section, and now you have sections that go all the way through the region instead of just looking at the first 5 sections. So you're right, Brady, that is biased. People are asking about steps before stereology. That is not in the scope of today's talk. Okay, I want to use unbiased stereology. This is from Chantel, to count area, to estimate area, volume, surface area of the lung, possibly also blood vessel length. If you go to stereology.info and type in lung, you'll find a section where there's a review of lung stereology, and it kind of goes through what I just went through, that you can estimate number, length, surface, and volume. You can do that in any organ, but do check out the lung section in stereology.info, because that will go through reviews that are just about lung. Will we have access to the video afterwards? Yes. How do you determine how often to sample slices? I'm going to get to that when we talk about precision. Okay, here's a good question from Krista. How can you best do systematic random sampling in human brain tissue when sometimes we do not have sections through the entire region? So everything I'm talking about is kind of a best case scenario, but it's true that with human brain tissue, we often don't have all of the region available. So the answer to that is do the best you can and write in the paper that you did not systematically and randomly section through the whole region. Okay, thanks for those questions. I really appreciate that. In the interest of time, I'm going to move on to the second category now, and that is sampling and imaging. So we've already talked about the fact that we need to use systematic random sampling. These sampling questions deal with other aspects such as imaging, and this first question from Charanjit from India, can we perform stereology on thin sections? And let's go back to stereology.info and just get reminded that the answer to your question is yes. If you are estimating volume, you can use thin physical or optical sections. If you're estimating surface or length, there are probes for thin sections, but then you have to manipulate your tissue blocks in a funny way in order to assure isotropy or randomness in 3 dimensions. So we really suggest that you use thick sections for estimating surface, estimating length, and for estimating number. So the answer is yes. Yes, we can use thin sections, especially for volume, for surface and length. Use thick sections, and also for number if you can. Katrina is interested in uses bright field and wide field, and the question is, do we have to use bright fields to image for unbiased stereology, or can we use fluorescence? The answer is yes, we can use fluorescence. Here is a typical system. This shows the hardware that you might be using that's connected to Stereo Investigator. There's a microscope and a camera. This is an apotome, which is structured illumination. That's a type of fluorescence that we can use. This controls the stage. We can't see, but in the back there's a bright field light source, so we can of course use bright field. There's a wide field fluorescent light source, and up here on the screen we can see that yes, we can use fluorescence to estimate the number of cells or do any of the probes. There's structured illumination here, and we also work with confocals. So the answer to that is yes, we can use fluorescence, we can use wide field, apotome, or confocal. Jordan from the US is asking, is it feasible and or reasonable to measure first order properties interested in cell number in tissue samples that are part of a tissue microarray? The answer to that is yes, you could use stereology in a 2mm punch, but keep in mind you'd only be finding out about that punch. Lucken from the United States writes, cost continues to be a limiting factor for the widespread application of stereology when dealing with preclinical pathology issues in drug development. What are some cost-effective aspects of your platform? So our platforms range from very cost-effective. In other words, you can get versions of Stereo Investigator that work with image stacks. We have Stereo Investigator Desktop, Stereo Investigator Whole Slide, and Stereo Investigator Clear Tissue. Those are all on the lower end of the spectrum of cost because they don't connect to hardware. If you've got a system that and all the hardware, that can run into the couple of hundreds of thousands of dollars. So there's quite a wide range. And if you contact us and tell us your specifics, we can help you with that. I do want to go to an example of using stereology, unbiased stereology, in pathology. So this is a probe called point sample intercept, and it's different than using the nucleator and the optical fractionator together because it won't give you a number-weighted estimate of volume. It will give you a volume-weighted estimate. And what that means is the way we sample makes it more likely that these hypertrophic nuclei will be picked if they're larger, because we put lines down and there's hash marks. If the hash mark goes through the nucleus, then the next step is to take that cross-section of the nucleus that's been picked for sampling because a hash mark goes through it, and then put two, in this case, red triangles on either side of the cross-section. And then that goes into a formula and gives you an estimate of the volume of these hypertrophied nuclei of cancer cells in the skin. So the answer is yes. We can use stereology for pathology. Keep in mind you're finding out just about what you're sampling. Joel from the US writes, please address how and whether we could use our neuroscience course MBF software to analyze our already acquired whole slide files, including pointers on acquiring the images for optimal stereology. Joel's interested in a workflow. So we do, you can use a slide scanner. We do have a product which is a slide scanner, but you could use any slide scanner. Joel's asking about a workflow, so it would have to be scanned and then stored someplace. These whole slides can be very large, these large montages. We have a product called BioLucida, which is great at storing these large image files. And then we have a version of Stereo Investigator that is not connected to hardware called Whole Slide Edition. And you can serve the large big data images directly to the Whole Slide Edition. So we do have a solution and a workflow for using whole slide files, getting them into our Whole Slide Edition, and then you could do something like estimating volume. Kevin from the US asks, writes, we're pursuing adding light sheet microscopy for clear tissue application to alleviate alignment issues on serial sections. Wouldn't mind seeing this modality in practice for NeuroLucida. Today's webinar is about Stereo Investigator, but I am going to show you an example of using a stack that is taken from cleared tissue, and that is in our program called Stereo Investigator for cleared tissue. I do also want to mention that we are working on our own light sheet microscope. It's called ClearScope, and it's coming soon. Here's a picture of it, and here's some images of what it can do. So keep in mind that we will soon have our own light sheet microscope available. And as I said, I'm going to, at the end of the third category, which is about estimating number, I'm going to show an example of doing the optical fractionator. Really now in this program it's called the volume image fractionator to estimate number of cells on a clear tissue stack. Igor asks, how can we do unbiased stereology with electron microscopy? That's an interesting question. Not many people are doing that. There's an extra consideration when doing electron microscopy when you're doing the systematic random sampling, and that's going from the microscopic level down to the electron microscope level. I'm going to show you a reference by Mark West where they did do systematic random sampling right down to the EM level. And if you take a look at that reference and get in contact with us, we can help you to do unbiased stereology on electron micrographs. Really, the bottom line here is if you can image it using systematic random sampling, then we can get it into our program and estimate number, length, surface, or volume. Next question is from Marina. how would one acquire z-stacks on a confocal for stereological estimates of cell count and size within a contoured area of tissue, for instance in the hippocampus, using Stereo Investigator? Can Stereo Investigator calculate co-localization of two markers or detection of one marker to another by proximity? Okay, so the answer is we have two ways of coupling with the confocal. If you're lucky enough to have our software on the same computer that controls the lasers, that controls your confocal, and you have a camera on that system, then that's a pretty easy coupling. You can use our program to do the systematic random sampling. When you get to the XY sites where your counting frame or dissector is, you switch over to the confocal program and take the confocal stack. That could be a multi-channel stack. Then we switch back over to our program to do the unbiased stereology. Most people are in a situation, though, where their confocal is multi-user. They don't have our software on there. So the other process you can use is to do this, figure out a way to do the systematic random sampling on the confocal, then bring those image stacks over to the computer that has our software. We have a module called the confocal module that will set up a special folder so that it can be seamless to do the optical fractionator once you have those stacks moved over to that system. Crystal from the US asks how to decide brightness adjustments when looking at these confocal stacks. And Alicia from Spain is asking about MRI scans. So I'm going to pause the PowerPoint and let's take a look at the three incarnations of Stereo Investigator that are made to accept images rather than be connected to hardware. That's what I'm using today. I'm at home. I'm not connected to any hardware. Stereo Investigator Desktop, you can load image stacks, confocal or bright field or any type. Stereo Investigator Whole Slide is made for the whole slide workflow from tissue scanner to BioLucida into Stereo Investigator Whole Slide for something like estimating volume using point counting. Stereo Investigator Clear Tissue, we're going to see an example of this today. This is set up to do probes instead of on physical sections, to do it on one very big section. I'm going to open Stereo Investigator Desktop and let's take a look at an MRI. Okay, I'm loading in the MRI image stack and I'm going to load it into one stack, and I can I put in the Z step, the X and the Y scaling is here. And now I can use my page up and page down keys to flip through this MRI. And a lot of people using MRI are interested in volume. And so we could definitely, here are the probes, number, length, surface, volume. We could go to the volume and do the cavillary estimator. When I hover over it, it says estimate region volume. You do that in thin tissue. So when I say OK, I get my points here, and it's very easy to mark points for volume in our program. So that's just an example of how, I'm not going to do the whole probe, but this shows that we can use MRI stacks, and we can do things like estimate volume. Now I'd like to open a confocal stack, because the question was, there was a question, can we use confocal for imaging? So I'm loading up a confocal image stack, and now I can use page up and page down to flip through this stack. I can go to image and show it at its actual size. And notice that this confocal stack has two channels, red and green. So there's some red fibers. We can turn those, we can look at one channel at a time. So I'm going to turn off the green channel. And now we're only looking at red. I can go to image and do a maximum projection. So this shows all the stacks together. And now I can adjust each channel individually so it looks its best. And you could use this to see if cells are double labeled or not. In this case, we could use it to see if there are fibers near cells. So we can open MRI stacks and confocal stacks. With the confocal stacks, it can be multi-channel. We can look at different channels individually and adjust the channels individually. So let's get back to our PowerPoint presentation. Okay, so let's go back to Marina's question. Her second question, can SI calculate co-localization of two markers? So you could use a multi-channel image to see if it's double labeled or not. Crystal asked how to define or decide the brightness adjustments, and we just saw that we can manipulate those brightness adjustments to get the image to look the way you want it to. Okay, so that is sampling and imaging. We saw that we can use thin sections for length and surface and number. We encourage you to use thick sections. And we saw that really any imaging modality that you can do the systematic random sampling with, we can get that into our program and do unbiased stereology. I'm doing pretty good for time here. I'd like to stop now, and if there are any questions that have to do with imaging, what type of modalities can be used, please type those now. Alicia Flores is thanking us for answering her question about MRI. They use fluorescent samples to use area fractionator. They have mono images. They have black and white images, and the question is it possible to add color to each marker? It sounds to me that if your markers are black and white, you may be running the program in mono mode. So that should be a routine thing to get colors on those markers. Okay, I have a question from Eric Castillo. What's considered thick tissue? For examples, in rats, we typically do 40 microns. So if you are sectioning them at 40 microns, and you're preparing the tissue for bright field, you're going to bring it through ascending alcohol concentrations, and it's going to shrink. So if it shrinks, say, from 40 to 30, that's thick enough in most cases, because you can use a 20 micron tall dissector and have 5 micron guard zones. So really it needs to be thick enough section to have a tall enough dissector so that you can have many focal planes through the dissector, and the guard zones need to be big enough to cover up any artifact. That's the theoretical answer. In practice, usually something like 30 microns is a good thickness. So as long as you're not shrinking down too much from 40 microns, you should be all set. Okay, Francois is asking, is it possible to quantify the density of fluorescence in a sample? That gets into things like relative optical density, and our program is not used for that. Charanjit is thanking me for addressing the question. You're welcome. Emily is interested in analyzing Z-stacks from the confocal. Should the stacks be saved a certain way, for example, as a .avi file? Our program can open many different types of image files, but really the best way is to save the stack and try it out and see if it can be opened in our program, and we can help you with that. Okay. I think in the interest of time, thanks for all these questions. We're going to move on to our last category today. So we've talked about using systematic random sampling. We talked about the fact that we can estimate number, length, surface, and volume. Those are all first order properties of tissue. Now let's get into the actual details of estimating number, because we have a lot of questions here, and this is by far what we got the most questions about compared to length, surface, and volume. There were a lot of questions about those too. But a lot of people need to, for instance, estimate how many cells are in a region. The first three questions have to do with how much sampling do we do? In other words, how do I set my precision? And when I look at the questions, these live questions, I'm getting a lot of questions peppered in there about how do I set my parameters? How do I know how many cells to count? Dumas from Turkey says, I'm interested in the number of objects. How many cells do I have to count to estimate, to get a good estimate of how many cells are there in my biological tissue? They know about the CE. That's a coefficient of error. It's a formula that tries to gauge the variance of your sampling. That is one of the ways how you're going to know if you've sampled enough. Connie's asked how to determine the counting frame area and the distance for each move. So the bigger question is, how do I know how many cells to count? How many cells do I count? Once you've kind of determined how many you count, the way you go about counting that many is by setting your dissector to be a certain size, by setting the spacing among the dissectors, and by setting the interval of the section. In other words, your parameters are going to affect how much you sample. So Connie's question is directly related to Dumas's question. Claudia says, hi, I'm a student that uses Olympus Stereo Investigator Bright Field Microscope, working on becoming a research doctor. I apologize if this seems like a silly question. This does not seem like a silly question to me, Claudia. It's very important. I'm unsure how accurate the stereology is in counting cells. So in other words, people may know how to count cells, but the question is, how many do we count? And that has to do with your sampling precision. Start in the literature. If it's like the hippocampus, you're going to find papers about it. For instance, if you are studying newly born cells in the dentate gyrus, I would point you towards Dan Peterson's papers, who uses the optical fractionator to do that and writes down his parameters and why he's using them. The second point here is you are the expert in your tissue. You know the salient features. For instance, if there's a clump of cells, so pay attention to the peaks of your data. If there's a clump of cells and you don't want to miss that clump, don't set your section intervals so that you could possibly miss it. It's good to talk to a mentor to find out why they are sectioning it the way they do. If you're really kind of lost about how many sections and how many dissectors to use to come up with your target of how many cells you want to count, there's a process called oversample and resample, where you can sample more than you think that you're going to need. And then this is a paper by Slomianka and West. It's a great paper to read. Then we've put this routine into our program where we resample. In other words, we ask, what if you had used half the sections? What if you had used a third of the sections? What if your counting frames had been spread out more? We have all that data. We can resample, show you what the estimates are, and you can find out if you can get away with less sampling and get the same precision. You're probably going to want to do a pilot study to find out things like the spacing of your dissectors and how big they should be. You're going to shoot for a couple hundred cells per region. There's formulas called CEs that you can look at. They gauge the variance from dissector to dissector or from section to section. And the lower the CE is, the less variance there is, meaning you're in a more homogeneous situation. Here is a way to set up a pilot study. Make your section intervals so you get about 10 sections. So if you're in CA1 of the hippocampus and you section all the way through that region and you get 100 sections, make your section interval 10, so you'll end up with 10 sections. Then on each section, set your counting frame, distances among the counting frame, we call that the grid step. Set that so you get about 10 counting frames per section. 10 sections, 10 counting frames. That will give you about 100 counting frames. Make the counting frames the right size to catch 2 or 3 cells. Some zeros are okay. More than 3 is okay. But we're trying to catch a few cells That will give you a pilot study with maybe 150 cells, and then you can look at the CE. Some labs have decided not to do that, and they just count a lot of cells. The point here is we can help you think about your precision and help you think about ways like the CE or oversampling and resampling so that you know the answer to these questions. How many cells do I count? How do I determine my parameters, which will affect that cell counting? How can I be sure that stereology is accurate? So I'm going to pause right here for questions about precision. Any questions about how many cells to count, how to know how many cells to count, how to know how big to make your dissector, how far apart to space them, how many sections. I'm going to just take a look at these questions coming in. Okay, this is from Chloe Page. This is a good question about coefficient of error. In the outputs of your program, there are 2 reports of CE. Which one should I use? What is the max acceptable CE? So there's actually, I think when you're saying there's 2 CEs, you're talking about the Gunderson CE, and there's 2 given, M equals 0 and M equals 1. One is more stringent than the other. If you're in doubt, just look at the higher CE. Those 2 CEs are there to differentiate between situations where you have a smooth transition between your cross-section, like maybe the amygdala, where the cross-section would be small, get bigger at the equator, and then get smaller as you section all the way through it. That would, you'd use the less stringent Gunderson CE for that. If you're in like CA1 of the hippocampus, your cross-sections are going to be big and then small and then big and then medium size and then big. For that, it's a more stringent CE we want to use for the Gunderson CE. There's also other types of CEs there. There's the Schmitz-Hoff CE and the Schaefer CE. The Schaefer CE looks at only the dissector and not at the sections. The Gunderson CE looks at sections and dissectors. So that's why you get more than 1 CE. And then this is a good question. What is the max acceptable CE? So we're not trying to think of the CEs as a hard and fast cutoff point, but traditionally people try to get their CEs down to 0.1 or below. If you're at 0.11, that's probably okay. If you're well below 0.1, then you might want to think about doing less sampling because maybe you can and get the same precision. Okay, thanks for your questions about how many cells to count. The bulk of the rest of these questions have to do with how do you do cell counting. Thomas is new to stereology. We'll be using this text soon for cell counting. Hisham says how to stereologically quantify neurons in a few brain sections to get an estimate for the total region. That's the whole point here. We're using a sub-volume, and if you are in a very small region, like maybe a subdivision of the hypothalamus, and there only is 3 sections, then yes, you use all the sections. But if you're in a region that has hundreds of sections, we use a section interval that will give us about 10 sections, at least for the pilot study. Draganya from Canada, use of optical dissectors for number of positive cells in the dorsal root ganglion. Again, any region of interest is fair game in any type of tissue. Shyam's interested in filament tracing. That's more for another webinar. We have a program called NeuroLucida that can trace processes in neurons. We are working on bringing a neuroscience forum to you. And in this forum, we could all communicate with each other about neuroscience, about things like neuron tracing and unbiased stereology and microscopy. We're excited about that. So look for our forum coming in the future. Shyam, that's where we could talk about something like neuron tracing. But counting microglia, that is very relevant for this topic. Sarah's asking, are there capabilities for assessing co-localization? We just saw that in the confocal image stack. And Sarah's interested in number of cells. So let's take a look at how to use the optical fractionator to estimate the number of cells. This can be done in thick sections. By thick sections, I mean something like 30 microns after histological shrinkage. You can orient the sections any way you want. You don't have to do that kind of strange manipulation that you'd have to do with thin sections for length or surface. We're going to use special rules, and we're going to go over those rules now, and we're going to use a virtual space called a dissector. We're going to look for the leading edge of the cell in the dissector to avoid bias. And of course, like all of these probes, we want to use systematic random sampling, both among the sections and within the sections. So this shows an image from our program. The red line is the tracing of the cross-section. The white dotted line is the grid space among the counting frames. The counting frames show red sides and green sides. Those are cross-sections through your dissectors. The warning on the bottom is not really a warning. It's telling you that you are not going to be able to control the throw of this grid down on the tissue. The only thing you control is the spacing. You control the systematic part. For each cross-section of your tissue, we throw this grid down randomly. That's why it's random systematic. Okay, how do we mark cells? Well, we don't want to be at low power doing this. I read a lot of optical fractionator papers, and the biggest mistake people are making is not using an oil lens, because we need to be at high power. Then we need to be in thick sections. We focus down through the dissector, and as soon as we focus on the top of the cell, we stop focusing and we decide whether to count that cell, whether it's in the dissector or not, based only on the cell top. This will normalize big cells to small cells, and it will take away the bias of orientation. So you can use this top of the cell. If it's a stellate-shaped cell, you may want to use the top of the nucleus. They're usually nice and oval. Stay away from widest extent of the cell or the nucleus. If there's only one nucleolus, you can use that nucleolus as the handle for the cell. We are dwindling the volume of the cell down to a point, so there won't be any bias due to the shape, size, or orientation of the cell. Okay, this shows cross sections. These are all cell tops, and this is a cross section through a dissector. It's called a counting frame. Notice it has a red side and a green side. If the cell top is in the counting frame, you count that cell as, but if the cell top is out of the counting frame, it does not get counted. If the cell top goes through a green line but not a red line, it gets counted. And on this funny shaped cell top here, we're showing that the red line trumps the green line. We want to use fixed sections, and we don't want to do this with a 10 times objective. We want to use an oil lens. That way we can get a thin Z-plane resolution. With the low power lens, here we can't even tell. These are 3 separate cells. We need to go up to an oil lens, focus through a thick section, going through the dissector, looking for the top of the cell, and then deciding whether it is in that virtual space called the dissector that we're taking a lot of pain to only sample cells in there. Okay, so the rules for the optical fractionator are: use systematic random sampling, use the leading edge or the top of the cell, do it with an oil lens and with fixed sections. If you follow all those rules, you can get an unbiased estimate of number. Let's go to stereology.info, and I have a link here to the page for optical fractionator. This shows the optical fractionator is to estimate number. If we go down here, it shows that it will estimate that in thick sections, and you can use preferred orientation. Let's take a look at the optical fractionator in action. Okay, so this represents a thick section, maybe 30 or 40 microns thick. That's the dissector. It would be something like 25 microns tall. And we can, I just made it transparent, but you won't see the whole dissector. You'll be focusing up and down through your thick section. And what you're doing is looking for the leading edge of the cell. We don't want to do this at low power and count the cell pieces. We want to do it at high power and find the top of the cell. Every cell has only one top, so we won't overcount the cells. I've just put a triangle on the top of the cell that I found by focusing up and down through the cell. There's its bottom, its middle, its top. And this shows that if I go back to the 3D view, every cell has only one top. If we can restrict ourselves to that volume fraction, the dissector, and we can be careful to count only the cell tops, we'll get an unbiased estimate. Okay, this is the formula for estimating the estimate. Q minus means how many cell tops you've counted in all the dissectors and all the sections. We take the reciprocal of the volume fraction. 4 things go into that volume fraction: section subfraction, area subfraction, height subfraction. If we're skipping every 10th section, we need to multiply by 10. If the ratio of the area sampled is 1 to 200, we need to multiply by 200. If you have a 20 micron tall dissector and 60 micron thick tissue, that's 1 to 3 for the z fraction or the height fraction. We need to multiply by 3. If we counted 100 cell tops in all our dissectors, in all our sections for this animal, then we get an estimate of 600,000. That estimate would be spot on if the cells were homogeneous, but they're not, and that's why you need to think about homogeneously distributed. So you need to think about how much sampling you're doing, which we already talked about, looking at the CE, doing oversample and resample. Okay, so right now I'd like to show a demonstration of the Optical Fractionator. Again, here is Stereo Investigator Desktop. This is Stereo Investigator for whole slide. We talked a little bit about a workflow from a slide scanner to BioLucida to serving big montages into this program to do things like estimate volume, or you could use point sample intercepts to get a volume weighted volume estimate of cells. But right now I want to open up Stereo Investigator Clear Tissue. Notice it's a counting frame inside of some clear tissue. And this is set up not for many physical sections, but for one big cleared piece of tissue. So I'm going to load that up right now. This is from the lab of Chip Gerfin, so thank you, Chip. This is a 1.5 millimeter chunk of tissue. We're seeing one cross section through it right now. I'm going to go to probes, and we still have number, length, surface, and volume, but when we look under number, I don't see optical fractionator. I see image volume fractionator workflow. So I'm going to start that. And right away, this workflow detects that this image stack is close to 1.5 millimeters. It's 1,436 microns tall. The first thing that it wants us to do is show us where the top of the region is. So I'm going to hit my control key and page down. And that's going 20 sections at a time. And I'm going to go just to where I think the top of this region is. And I'm going to click Set Top of Region. So that's this right here in this diagram. I just set the top of the region. Now it wants to know the bottom of the region. So I'm going to focus all the way down to the bottom of this huge image stack. It's not updating as I go. It will update when I get to the bottom. Okay, so I'm at the bottom of the image stack. It looks like this. And I'm just going to say that that is the bottom of our region. So set bottom of region. So I just defined from minus 91 to minus 1,433. That's where I want to sample. That's our region. The next thing that I'm going to do is break this region up between this and this into virtual sections. That's the whole point of this clear tissue using light sheet microscopes, is that we don't have to deal with physical sections, with many physical sections. We just have one. So we need to make virtual sections in here. If I made 5 virtual sections, it's going to take this distance from the top to the bottom and break it up into 5 virtual sections, and those happen to be 286 microns. So I'm going to have 5 virtual sections, 268 microns tall. I'm going to make a dissector height of 100 microns, and then click Next Step. And it populates over here in the Serial Section Manager, 5 virtual sections. It brings me to a random place in the middle of the first virtual section. And now my job is to trace the region of interest. I can focus up and down a little bit to see how big that region might be in this virtual section. Now I'm going to go ahead and trace that region. And then go to Next Step. It's going to bring me to 100% zoom level. These white dots are the cross sections through the cells that we're estimating. This counting frame is 100 by 100. I think that'll be a good size to catch a handful of cells. We define the systematic random sampling layout. I'm just going to use something appropriate for this demonstration. We can save the sampling parameters. Now I'm on Count Objects. I click on this contour and I do Start Counting. It brings me to the first systematic random location, and it gives me a Z-map of my 100 micron tall dissector. And now I start focusing down. I don't see any white dots in there right now. So I'm focusing down through the Z, and I'm looking to see if a cell top comes into focus. And I can see a cell top coming into focus. I can see 2 cell tops coming into focus. So I'm going to mark this cell top and this cell top. Okay, I'm not going to spend a lot of time marking these cells. You just saw 2 examples of that. What I want to do is get to the next section, because this is really the beauty of using clear tissue. So I'm done with one section. I would have gone through each one of those, focusing down, marking cells. It would take me something like 20 minutes. So I say, okay. And now it's time to go to the next section. Prompts me to save, which is good. It's time to go to the next section, but the beauty here is I don't have to take a physical section off the microscope and find the other physical section and then get everything in focus again. I'm dealing with virtual sections on one huge image stack. So I just do Begin Next Section. It's telling me you're going to need to trace contours here. It brought me, again, it's telling me to trace contours. It brought me to a random place in the middle of virtual section 2. So once we start getting a lot of these clear tissue image stacks, it's really going to revolutionize the way we do the optical fractionator. Okay, so that's a demonstration of the optical fractionator to estimate the number of cells using Stereo Investigator clear tissue. Let's go back to the PowerPoint. We answered these 3 questions about precision, and now we've shown how to use unbiased stereology, specifically the optical fractionator. You could do that with a live image. You could do that with captured images using the desktop version. But what I showed was clear tissue version of Stereo Investigator. I'd like to end with this last question from Ayon. I've been using ImageJ software for quantifying cells from my data. I find the method very subjective. That's because it is very subjective. you're probably dealing with not a three-dimensional situation, just counting cell pieces. This is why we really want to use unbiased stereology. We want to use the optical fractionator to come out in a sound and scientific manner to get an unbiased estimate of the number of cells. Okay, I know that the hour is up, but if anybody, people who are still with us, let's pause right now for questions about estimating number. Okay, when dealing with clear tissue, this is from Renee Oliver. When dealing with clear tissue, light sheet microscopy data, the files are normally really big. How well does the software deal with this data, and which hardware is recommended? So we're all facing this. TIFFs are getting bigger and bigger. You can use JPEGs, which are compressed. Those are good to use, but those themselves are pretty big. We have a version of our software coming out, especially with our program NeuroLucida 360, which is going to really do much better handling these big versions. I just meant to say it will be in NL360 first. It will be in all our products eventually. But right now, you're right. We now have the capability to image these huge clear tissue chunks, which give us really big image stacks, and one limiting step may be having enough memory to deal with those. But relief is coming, and we're going to have a better way to deal with big images. Let's see. Okay, where are we on the possibility of automated counting? I'm just going to say we are working on that. And Shirley says, thank you for this. Will you have a webinar on neuroscience stereology estimating volume? We definitely have enough questions. Here are questions that I didn't get to. We have questions on length. We have questions on volume and cross-sectional area. We have questions on other topics like global versus local probes, second order stereology, histology analysis. We do have enough questions for at least another webinar, and I would like to give another one of these. I think it was very successful. So now, please send in questions on any category you want. We've gone over stereology overview, sampling and imaging, estimating number. There are some suggestions for other topics or any category that you want to ask a question on. Send those in now. I'd like to again mention that we're working on a neuroscience forum where we'll all be able to talk with each other not only about unbiased stereology for estimating number, length, surface, and volume in neuroscience tissue, but also tracing neurons and microscopy. So I'm excited about that, and so look for that forum in the near future. And thank you very much for joining us today. This it was recorded and will be available. And there is the website to contact us about anything. Thank you.